Very excited to be here, and I think this is a very exciting initiative to try to understand the brain more, because I am a neurologist, and as Danny said, we do have some diseases that are challenging to treat. We do have, of course, a lot of other ones where we make a major, major difference in their patients' lives, but there are some, and part of what draws us to this field is that there's so much promise for advancements. I mean, we really can make huge substantive differences in individuals' lives with the research that we apply and the research that we're going to be talking about today. Hopefully I'll leave you with a little bit more optimistic viewpoint of some of these illnesses because of what we're learning in the brain and some of the tools that we're building. So I want to talk to you a little bit about successful aging and brain structure changes in aging and disease and brain injury. But my focus a lot is using some of these new technologies to actually learn more about the function and structure of the brain. So I'm going to talk to you first just somewhat simply about measuring brain structure with MRI in a quantitative fashion, which shockingly has not been applied in the current practice of neurology until very recently. And so I've really been trying to push for more quantitative approaches in analyzing brain structure, because we as neurologists are all about making structure-function relationships. So if we don't know sort of what is going on with the structure and that's not getting fed back to the neurologist, then they're really hamstrung in making those structure-function relationships. So a lot of times what I'll show you is the radiology communication that we make between the referring physician and the radiologist. There's a lot of loss of communication because the radiologist will try to translate that brain image that they're seeing into a single paragraph. And so you lose a lot of data in that transport. And so I'm talking about using technologies as we're discussing developing here in order to improve that bridge in communication. And I'll show you that in more detail. The other exciting advances that are really helping our clinical trials in specifically Alzheimer's disease, but also in brain injury, are the ability to now view amyloid, or evidence of buildup of amyloid, which is a protein that builds up in Alzheimer's disease, one of the bad proteins. And the other bad protein is tau. And tau is very, very prevalent in brain injury. And so these are very exciting new tools, and I'll show you just a taste of that new technology that's really changing how we do trials. And then also some other research horizons that hopefully our work with the Brain Initiative will allow us to get more accurate and quantitative measures of brain activity with MRI and also positron emission tomography, or PET scanning. And probably won't have too much time to get into the restriction spectrum imaging, but there are some very exciting physics types of advances that one can apply with magnetic resonance imaging, which are really, really exciting. And I'll try to get to it, but we're running behind, so I'll work on this. So first thing I want to just tell you about is why are we hearing so much about Alzheimer's these days? It didn't seem, is there a greater prevalence of it? Well, indeed there is, but it's really a function of the fact that our population is growing older because we're able to stop many of the illnesses that used to kill you when you were younger, right? So heart disease and cancer, there's many new advances that otherwise would have not been available to people and having those illnesses. And so you see on the left side what the old state was when we didn't have those treatments, and you'd see that very few people would make it into even the 85 years of age. Whereas now we have what we call a population pillar. That is, very few people are dying at these younger ages, and so all of them are making it into these elderly ages where we're seeing diseases that we have much more trouble taking care of, like Alzheimer's disease. So centenarians means a person who's lived to the age of 100, and that was quite rare in the past. In 1998, it was only 61,000, and it's expected that by 2050, there'll be 600,000 that can live into that age. And now let's go even further to supercentenarians. Those are individuals that live past 110, and you'll see that that is extremely rare because across the world and recorded, you see that past age 116, which is quite rare to achieve, you have almost all of it accomplished by this world record holder, Jean Calment from France, who lived to age 122 almost. And and actually quite spry in in her interviews. Even at that age, she would say, "Well, I think God just forgot about me," or, she would say, "Well, I have one wrinkle and I'm sitting on it." So her brain was actually quite well functioning, and so we may learn a lot from those healthy, successful cognitive agers, and that's. In fact, I'll talk to you about one of the new drugs for Alzheimer's disease that is getting a lot of excitement in the field that really was built based on taking healthy cognitive agers and seeing what is it about them that's allowing them to maintain brain function into their 90s and beyond. And then saying, well, we found something that may be special as a target, and then trying to mimic that molecule and actually putting it into trials. And that drug, which we'll talk about, is actually going to go straight from phase 1, which is a very early stage, all the way to phase 3, with skipping phase 2 because of the results they saw in phase 1. That is, they had so much excitement about the phase 1 early stage studies that they said, we're ready to invest and risk $1 billion on this drug because we're so optimistic that this drug is going to have its effect. So studying the healthy cognitive agers is something that's quite useful, and we do it here at the Shiley Marcos Alzheimer's Disease Research Center, where people can come to volunteer for our studies, and we have a number of different studies that are quite exciting. So what are some of the things that you see even when you're aging in a relatively healthy way? Well, regardless, even 83% of people that are in the 70 or above will say, well, I'm having some trouble recalling names or words. You say, well, that's just normal. Well, it's normal, but let's kind of figure out, well, what about those 17% that's not even having that kind of a complaint? Is there something special about that? But other common complaints of recalling where you are, where you put things, knowing you told someone something, what was I just doing, when I walked into this room, and losing the thread of a conversation. These are common complaints that we hear in the neurology clinic, and the big trick is to say, well, which of these are concerning and which of these are not, because a lot of people are complaining about these, and some of them will just go on and age quite successfully. So these are some of the changes that occur as one person is aging. So normal aging, of course, the brain slows down, and you'll get some of those complaints that we just saw on the prior slide. Medications, of course, as you build up medications even for things like heart or bladder function, those can affect your cognition and memory. Alcohol, depression. Depression can be so severely affecting of memory that it can be called pseudo-dementia. Very treatable, looks just like dementia, but the person is just severely, severely depressed. Anxiety itself, even having a family member who's had Alzheimer's disease, has gone through the trauma of that experience, and then as that patient gets to that age and starts to have normal slips in memory, like parking the car in a spot and saying, wait a second, where did I park that car, or losing the keys. For you and I, a lot of times that would not be too distressing, but for a person who's really dealt personally with the effects of a family member with Alzheimer's disease, it can be absolutely devastating and huge anxiety, which in fact, if we can get a better sense of whether this is concerning or not, we can actually relieve a lot of that stress and anxiety by really having a better test to say, well, is this something that is going to move on and get worse, or is this going to stay stable? That's what the patients are asking us for, and that's what we'll talk about today, about trying to develop. Sleep problems, and then, of course, Alzheimer's disease and other neurodegenerative disorders, which are quite common and shockingly common, in fact. So let's talk a little bit about Alzheimer's, which is the most common cause of dementia. It is dementia itself is an umbrella term that encompasses more than just Alzheimer's disease, but Alzheimer's is the most common of dementias. And so it is causing degeneration of the neurons, and you'll see memory loss as its hallmark. About 5 million people in the U.S., but that's really people with Alzheimer's dementia. That means at the latest stages of the illness. We're now learning that this illness starts 15 to 20 years before a person reaches the stage of Alzheimer's dementia. And so we're talking about many, many, many people living with the initial signs of memory loss due to Alzheimer's disease. It does increase with age, but greater than 40% prevalence at age 85. That's almost a coin flip. If you live to age 85, that you will be suffering from actual dementia due to Alzheimer's disease, which is a shocking statistic. It means that we really need to get on top of this illness, or else it will devastate our health system. So what is my passion in neuroscience? It's a medial temporal lobe, and it relates to Alzheimer's disease because this portion of the brain, which I'll talk to you about, is critical for forming new memories. That is, if you want to lay down a new memory about the experience or even today's talk, you're using a structure called the hippocampus very, very extensively to lay down every trace of memory. And this is damaged in trauma, stroke, epilepsy, and Alzheimer's disease and other neurodegenerative illnesses. Alzheimer's is probably your most salient one because you know that as you age and as memory starts to slip, the first concern is, well, geez, is this the first sign of Alzheimer's disease? So knowing that we want to do structure-function relationship, let's look at the kind of changes that occur in the brain that relate to neurodegenerative illnesses such as Alzheimer's disease. This structure that you just saw expand, this is actually a fluid-filled structure that is right next to the hippocampus. So we see that fluid-filled structure start to expand as you lose tissue in this area, and it's actually pretty, if you've looked at enough Alzheimer's brain scans, just structurally, you can say, boy, that looks a lot like Alzheimer's disease. The only problem is that our radiology colleagues, they see all kinds of scans coming in, and they don't know how much is normal shrinkage with just regular aging and how much is actually a neurodegenerative cause of shrinkage. So that's what we'll talk about in just a moment here. This is a dissection I did while I was a student at Sanford, where I was very passionate even then about the hippocampal function. So I left the hippocampus intact. And you can see it's just a pretty small structure, but it is critically, critically important for laying down memories. And then this is a big fiber bundle. And Terry talked about some of the diffusion tensor imaging, and you can really pick this up with some sorts of magnetic resonance imaging that I'll try to get to. So this small structure is what we're really focused on, and it's called the hippocampus because hippocampus means seahorse, and this is a very small structure. Again, my hand holding the hippocampus, and it's thought to look a little bit like a seahorse, but I try to help you out by sticking the seahorse on there. But so that's the hippocampus, and that's kind of the theme of today's talk. It's really an amazing structure that differs in its cytoarchitecture and connectivity based on where you're slicing it in the brain. And we learned a heck of a lot about it from a unfortunate patient in 1953 who was suffering from seizures. He was having about 12 seizures a day and took the medications at the time, which were phenytoin and phenobarbital, but it wasn't stopping his seizures. He was in his 20s and said, I can't live like this. I can't have 12 seizures a day and hold down a job, so I'll do anything to try to stop these seizures. And he went to Hartford Hospital in Connecticut, and they said, well, we've mapped the seizures. It looks like they're coming from your hippocampus, so let's do a surgical procedure in 1953 to remove both of your hippocampi. And from that day forward, that unfortunate patient, Henry Molaison, or HM, couldn't form no new memories, even things as salient as his own mother dying. As soon as he was distracted from that information, it was as if you were saying it again. Man landing on the moon, be completely shocked and amazed like the rest of us about that information, but as soon as he's distracted from it, you tell him again, and he's just as shocked and amazed as the first time because you're unable to lay down that new memory. He was actually quite intelligent. He just didn't have memory, so he could describe what's going on. What are you feeling? He says, well, right now I'm wondering, have I done or said anything amiss? See, at this moment, everything looks clear to me, but what happened just before? That's what worries me. It's like waking from a dream. I just don't remember. And so I think anybody that's dealt with patients with Alzheimer's disease, before the disease has spread far beyond the hippocampus, it is kind of that state of every moment is it stands alone and very difficult to relate to prior experiences. We'll be testing patients and we'll test their memory, and you see them start to get very upset about the loss of memory and how they're doing on the test, but with a quick distraction, all of a sudden they've moved on, and that distress and upset is gone. So in some ways, a little bit merciful, but otherwise you see it as just an awful thing because, what makes up our life and our richness is our memories. And so this is something I've had a passion in trying to work toward since before medical school. And in fact, it drove my motivation to go to medical school, is to really work on memory and Alzheimer's disease. I was working in brain imaging of patients with Alzheimer's disease in Berkeley in 1990, and so I was the guy kind of trying to help keep them still in the scanner. So I interfaced with the patient a lot, and since I was just the kid there holding their hand and trying to keep them still, they often told me their stories, and I was just struck by how devastating this illness is. So that was in 1990, and it took 40 minutes to get 12 slices through the brain in an MRI scan, and it was basically, just the initial usage of high-resolution imaging. Before that, we had CT scan, which gave you kind of a fuzzy picture, but nowadays we have a very beautiful, almost like you're cutting open the head and looking directly at the structure of the brain. We now have exquisite pictures of anatomy that can be applied in a quantitative manner to really look at anatomical change that takes place. And why is that important? Well, let's just look at a brain like this, where we have a very nice-looking back part of the brain called the parietal lobe, but you move forward and you see these nice, thick, what we call gyri and folds. You go up into the frontal lobe and you see severe atrophy. And I don't know if you see it, but I'll compare it here to a different brain. You see a large, nice gyri in the back, but very shrunken gyri in the frontal lobe. This is called frontotemporal dementia, a different type of dementia. You can compare it to, I guess, a little bit of self-promotion. This is my own brain, 3D printed. So with MRI, you can actually take the anatomy and send it to a 3D printer. It's kind of cool. You don't have to die to hold your own brain in your hand. As far as I know, doesn't have frontotemporal dementia, so the comparison is, decently full gyri and very shrunken gyri. Using that technology, I thought, wow, that is so cool to have your own brain holding in your hand. So we had a group of X Prize donors. These are a lot of them multi-billionaires who came down, and I wanted them to get excited about neuroscience because we always are looking for people to be movers and shakers and trying to move this field forward. So I 3D printed all their brains, and I had, so I did 37 of these brains for X Prize donors, but the point here is that each individual's brain has subtle differences, and as they age, they will degenerate in different ways. And I think we can use these kind of quantitative approaches to really detect that. We can look at the brain as if you took it out at autopsy, but the person's still alive, and you have a snapshot when this person was in their 40s, when they were in their 50s, when they were in their 60s, and they get the idea. Like, you can have it on your shelf and say, this is how my brain has evolved over these past 3 decades. And I tell them, well, okay, you get the idea, but the truth is you don't need to 3D print it to get that quantitative information. In fact, the computer is better at detecting the subtle changes that are taking place across time. You don't have to wait 10 years. We can see these subtle changes within 3 months and see whether the person is actually atrophying at a rate that looks beyond what they should be atrophying. So what I'll talk to you about are the quantitative approaches we've pushed forward to try to bring that into clinical trials to help us better understand whether a drug is having an effect or not. Is this drug actually slowing the neurodegenerative process? Is it sparing brain tissue after a brain injury? What can we do to improve that? Well, we want to provide a referring physician with quantitative information extracted from the images. This is not new. Anybody who's gone for an echocardiogram knows that you get quantitative information about how the heart is functioning. Tell me what your ejection fraction is. How much of that blood is being pushed out of that left ventricle every time the heart beats? And if you didn't get that quantitative information, you'd be very upset with your cardiologist saying, I need to know what it is now relative to my old measure. I'm arguing that we should be able to do that also with not only our PET scans, but also with our MRIs. So just a quick flip through this, just to show you that we now have fully automated approaches to get the kind of data that used to be required through hand drawing of the hippocampus in the 90s. We now can get in 5 minutes with advanced computer processing and computer vision techniques. So this allows us then to do this on thousands of individuals because it only takes 5 minutes and it's a button press. So once you can do it on 1,000 well-characterized individuals, you basically have a very nice normative database to say, well, what is the normal volume of the hippocampus for a healthy 84-year-old person? What is it for a healthy 79-year-old female? So, and now you say, well, where is mine relative to those other numbers that you've collected? So this is the type of base procedure that we do. We get a high-resolution 3D scan of the brain. We quantitate it. We measure it, and you can see just qualitatively looking at this, the change that happens between a healthy and an Alzheimer's patient. So that should be detectable, right? But whenever it's a gradation like that in severity, it's difficult for a radiologist to really say, well, how much is too much? Okay, so when you put it right next to a nice healthy brain, you go, well, clearly that's too much. But so what we want to do is really to be able to use that imaging phenotype to change our probabilities and be able to give more information to these individuals about what's going to happen. Because then you also can enroll them in clinical trials much more effectively to know that that individual is going to decline during the next 3 years. And now that's the person you really want to intervene on and see whether the drug takes effect. One of the reasons a lot of these prior trials have failed is we've enrolled a mixture, a bag of elderly individuals who are complaining of cognitive complaints, and we put them into the trial, but not all of them have Alzheimer's disease. In fact, we now have the number, basically, almost 1 out of 5 patients that were in prior Alzheimer's trials didn't have Alzheimer's at all. So that, of course, is going to mix up your results and make your trial much less powerful. So that's what we're using biomarker. Luckily, Sanjay Gupta liked our work, and that got us a little bit of attention. We've been able to really predict in almost a very clear manner whether an individual who has cognitive complaint is going to progress or stay stable. This is now guidelines that are hopefully going to be more widely adopted because the current criteria for diagnosing Alzheimer's disease in the clinic were developed in 1984. And so there's a big push to say, why are we still using those old criteria? But medicine moves in a very conservative manner. I say, this is how I was trained, so I'm not changing it. Well, there's a push amongst key opinion leaders to say, no, we should use these biomarkers in clinical practice. All right, so I think I'm just going to jump right up to amyloid imaging because that's something I think is quite relevant in this talk here, because it is a major advance that's going to push forward our understanding of this disease and also of brain injury. So in a healthy brain, not only would we see the healthy brain structure, but we don't see this amyloid protein buildup that you see in an Alzheimer's patient. This is an atrophied brain with Alzheimer's disease, and you see all this brown stuff. That's a bad protein that's being aggregated in the parenchyma of the brain or the tissue of the brain. Now, we used to only be able to get this information after the person died, and that's not very effective in clinical practice or in clinical trials. So it's very, very important to be able to see this in the living individual, and now we're able to. We have 3 agents that are actually FDA cleared now and available, although not covered by Medicare or by other insurance at this time, because there's a lot of devil in the details of what this means to have amyloid buildup in the brain for the diagnostic process. But these individuals with Alzheimer's disease have clear buildup of amyloid in the brain. The blessing and the curse of it is that it's an early biomarker. I always say the blessing is the same as the curse, right? Because if I tell you you have amyloid in your brain, and I don't know whether you're going to have actual cognitive complaint due to that amyloid for another 20 years, then it's not that helpful at this time to have told you that you've had amyloid in your brain. All I've done is taken a worried well patient and turned them into a devastatedly worried patient, okay? So that's why I think the use of this in clinical practice is very touchy, and I agree that we should be very conservative in applying this in clinical practice. However, for clinical trials, it's absolutely clear. You want to know as a researcher, does my patient have the target protein that I'm trying to attack? And so that's what we now have, and we have ways to quantitate it. And then finally, I'll wrap up on the ability to actually see the other bad protein. This is hot off the presses. In fact, these are the first images of patients with Down syndrome, which we could get into. As patients with Down syndrome, as they age, they have extra amyloid protein because they have 3 chromosome 21s. So chromosome 21 has the amyloid precursor protein. So they produce 150% of what the rest of us produce in that bad protein. And so all of them, if they live long enough, will develop Alzheimer's disease pathology in the brain. And so we did a study with, this is a patient in their 30s, this is a patient in their 40s, and this is a patient in their 50s. And this patient had a genetic background that also had a particular genetic risk for Alzheimer's buildup. So in fact, his tau protein was higher than this older patient because there is an interaction between all the genes and the aging process. So these are the very first images of the bright tau that builds up in the protein in the brain of patients who have Alzheimer's disease and also brain injury. We can now look at it in the living individual, and I just argue that this is going to be extremely revolutionary in our ability to detect and build on the processes of disease, not only in Alzheimer's disease, but in brain injury. So being able to actually quantitate this and be able to show that a drug is having an effect on the trajectory of the buildup is going to really revolutionize the way we do trials. And so that's only a little taste. I'm super excited. It's very hard to stop me at this time, but I know we're behind. So at that point, I'll just stop here, and thanks very much for your time.